Hello, everyone, and welcome back to the lecture series on synthetic and biological macromolecules. Gregor Mendel, who carefully studied more than 8,000 pea plants, was the first to recognize that specific characteristics were passed from one generation to the next. He published his findings in 1866, but it was not until 40 years later that the term gene was coined. At the time, proteins were believed to be the repository of inherited information until the 1940s, when experiments concluded that deoxyribonucleic acid, commonly abbreviated as DNA, was fundamental to the process. Many researchers contributed vital information that eventually led James Watson and Francis Crick to propose the structure of DNA in 1953. DNA itself was discovered in 1869 by Friedrich Miescher, while the Russian-born biochemist Phoebus Levine determined its component parts, a sugar, a phosphate group, and 4 nitrogenous heterocyclic compounds, referred to as bases. In this lecture, you will be introduced to one of the most remarkable known biopolymers. Hopefully, by the end of the course, you will have a general understanding of the components and function of DNA. Life begins with nucleic acids, which are unquestionably top-level molecules as they store genetic information. Nucleic acids are polymers whose building blocks are nucleotides, each consisting of 3 parts: a heterocyclic base, a sugar, and a phosphate ester. In the example presented here, adenine is the base. The combination of a base with a sugar is called a nucleoside, and in this case, the nucleoside is known as adenosine. When a phosphate group is added to the nucleoside, the resulting molecule is called a nucleotide. This particular nucleotide is known as AMP, adenosine monophosphate. Nucleic acids can be classified into 2 major groups: ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. The 2 types of nucleic acids differ in the sugar portion of their mononucleotides. RNA contains ribose, a 5-carbon sugar, while DNA contains deoxyribose, in which a hydrogen atom replaces the hydroxyl group at the 2' carbon of sugar. In nucleic acids, only 5 bases, 2 sugars, and 1 phosphate group are possible. The bases are either monocyclic pyrimidines or bicyclic purines, and they are all aromatic. More specifically, there are only 2 purine bases found in nucleic acids. We have already encountered adenine, abbreviated as A. The second purine base is guanine, commonly denoted as G. The 3 pyrimidine bases are uracil, thymine, and cytosine, which are structurally simpler. Pyrimidine bases are abbreviated as U, T, and C, respectively. Cytosine is found in both DNA and RNA. Uracil is found only in RNA, and thymine is present only in DNA. One of the most important discoveries of modern science was the elucidation of the structure of DNA as the famous double helix by Watson and Crick in 1953. They recognized that the base sugar phosphate framework was ideal for forming a 3-dimensional coil. A small portion of one of the DNA helices is shown on the right. Notice that the 2' position on the ribose ring is vacant. There is no OH group there. This absence is why the molecule is called deoxyribonucleic acid. The nucleotides are linked through the 2 remaining OH groups on the ribose ring, located at the 3' and 5' positions. The small DNA fragment shown here consists of 3 nucleotides: adenine, adenine, and thymine, and is abbreviated as AAT. In the cell nucleus, DNA exists as 2 chains wrapped around each other in a double helix. The polar sugar-phosphate backbone of each chain interacts with water in the cellular fluid via ion dipole forces and hydrogen bonding. In the core of the DNA, the nonpolar bases form intrachain hydrogen bonds, which hold the 2 strands together. A double helical DNA molecule may contain millions of hydrogen-bonded base pairs. There are two key features of these base pairs that are crucial to the structure and function of DNA. First, a pyrimidine always pairs with a purine, ensuring that the double helix maintains a constant diameter. Second, in healthy cells, each base is always paired with the same partner. Adenine always pairs with thymine, and guanine always pairs with cytosine. In other words, the base sequence of one strand is the complement of the sequence on the other. For example, the sequence adenine-guanine-guanine on one strand is always paired with the sequence thymine-cytosine-cytosine on the complementary strand, as shown in the middle of this figure. Another important feature of hydrogen bonding is that adenine-thymine pairs are held together by two hydrogen bonds, while guanine-cytosine pairs are stabilized by three hydrogen bonds. These hydrogen bonds come in two types. One links an amine group to a carbonyl group, and the other connects an amine group to the imine-like nitrogen in the heterocycle. In this way, each nucleotide reliably recognizes and pairs with its specific partner. Segments of the DNA chains form genes, which contain the chemical information necessary for synthesizing the organism's proteins. Each DNA molecule is tightly packed into a tangled mass that forms one of the cell's chromosomes. A DNA molecule is astonishingly long and thin. If the largest human chromosome were fully stretched out, it would be 4 centimeters long. In the cell nucleus, however, it is wound into a structure only 5 nanometers in diameter, 8 million times shorter. An amazing feature of biopolymers like nucleic acids is that they can copy themselves. This process involves a complex series of interactions and is called DNA replication. When a cell divides, its chromosomes are replicated, ensuring that the new cells have the same number and types of genes. During replication, a small portion of the double helix is unzipped, allowing each DNA strand to act as a template for a new strand. Then, the bases in each DNA strand form hydrogen bonds with the complementary bases of free mononucleoside triphosphate units. In the final step, the nitrogenous bases, stabilized by hydrogen bonding, are linked through phosphodiester bonds to form a new strand. This process is catalyzed by an enzyme called DNA polymerase. Gradually, each of the unzipped strands forms the complementary half of a double helix, resulting in 2 identical double helices. Since each base always pairs with its complement, the original double helix is faithfully copied, ensuring the genetic makeup of the cell is preserved. Astonishingly, this complex process relies heavily on hydrogen bonding between complementary base pairs. The information content of a gene resides in its base sequence. In the genetic code, each base acts as a letter. Each 3 base sequence functions as a word, and each word codes for a specific amino acid. For example, the sequence cytosine-adenine-cytosine codes for the amino acid histidine, while adenine-guanine codes for lysine, and so on. Through an astonishingly complex series of interactions, amino acids are positioned one at a time and linked together to synthesize a protein. To fully appreciate this fundamental aspect of biochemistry, keep in mind that the process relies heavily on hydrogen bonding between base pairs. So, protein synthesis is conventionally divided into 2 main stages: transcription and translation. DNA is located in the cell nucleus, but the genetic message is decoded in the cytoplasm. Therefore, the information must be transmitted from DNA to the site of protein synthesis. RNA serves as this messenger. A highly simplified representation of this process is shown in the accompanying figure. Transcription produces messenger RNA, which is a copy of the DNA information. That is, the information is transcribed from one nucleic acid to another. Transcription shares some similarities with DNA replication and begins with the temporary unwinding of a portion of the DNA. One strand of DNA serves as a template for the synthesis of a complementary messenger RNA strand, which is formed by linking individual mononucleoside triphosphates. The DNA codons are transcribed into RNA codons through base pairing. Cytosine and guanine pair as complementary bases in both DNA and RNA, while in RNA, thymine is replaced by uracil. Following transcription, the DNA rewinds, and the messenger RNA exits the nucleus through pores in the nuclear membrane to initiate the next stage of protein synthesis, translation. Translation uses messenger RNA to synthesize proteins. That is, the genetic message is translated from the language of nucleotides to that of amino acids. Translation begins when messenger RNA binds to a ribosome, an RNA-rich particle in the cell. This process again relies on base pairing via hydrogen bonding. Next, transfer RNA molecules bring amino acids to the messenger RNA bound to the ribosome. Transfer RNAs are smaller nucleic acids, represented here in brown. Each transfer RNA molecule has two key regions. One end contains a 3-base sequence known as the anticodon, which is complementary to a specific 3-base codon on the messenger RNA. On the opposite end, there's a covalent binding site for the amino acid that corresponds to that codon. Amino acids are depicted here in different shapes, colored red, blue, and purple. The transfer RNA then shuttles the correct amino acid to its corresponding codon on the messenger RNA. As the ribosome moves along the messenger RNA strand, one 3-base codon at a time, each transfer RNA molecule binds to the messenger RNA via hydrogen bonding, positioning its amino acid in sequence. Once a transfer RNA molecule binds to the messenger RNA at the ribosome, it releases its amino acid, and an enzyme catalyzes the formation of a peptide bond, linking the amino acid to the growing polypeptide chain. Finally, once its amino acid has been released, the empty transfer RNA exits the ribosome and can be reused in another round of protein synthesis. In essence, protein synthesis involves transcribing the DNA codons into an RNA message and then translating that message via transfer RNA molecules into a sequence of linked amino acids that form a protein. To sum up, in this lecture, you have learned about the nucleic acids DNA and RNA. You saw that these biopolymers are polynucleotides, consisting of 4 different types of mononucleotides. These mononucleotides are based on nitrogen-containing heterocycles: adenine, guanine, thymine, and cytosine. In RNA, thymine is replaced with uracil. These nitrogen-containing bases are linked together into long chains via sugar and phosphate subunits, forming polynucleotides that are, in essence, polyesters. The 2 strands of DNA form a double helix when complementary nitrogen bases pair through hydrogen bonding. Adenine pairs with thymine via 2 hydrogen bonds, while guanine and cytosine are held together by 3 hydrogen bonds. You have also seen that the base sequence of the DNA strand determines the amino acid sequence in proteins. Hydrogen bonding between specific base pairs is the key to both protein synthesis and DNA replication. In the next lecture, we will begin examining synthetic macromolecules. Thank you for your attention.